bit of a systems check. Looks like everything is going okay. And let's go ahead and do the second part of the PowerPoint. And let's pick up where we left off. We were starting to talk about the second line of defense. So keep in mind that the first line of defense, that was the skin and all the mucous membranes. So the second line of defense triggered when body tissues are injured, redness, heat, swelling, results in a chain of events leading to protection and healing. So the inflammatory response is always very important. That means that your body is working well enough to be able to fight what has gotten into your skin. Any kind of cut, or again any kind of maybe a cut, a paper cut of some sort, prevents the spread of damaging agents, disposes of cellular debris and pathogens, sets the stage for repair. So inflammation on an area, sometimes you might get cut and see that, it gets kind of red surrounding the cut. That's called inflammation. That is a good thing, okay? That means that your body is truly doing what it's supposed to be doing and fighting off any kind of bacteria that might be getting into your skin. There is also something called complement, a group of at least 20 plasma proteins. And this complement, what happens is these proteins will get together and actually form kind of a ring inside a cell membrane. And a lot of times, I know this looks kind of weird, but actually the complement gets into the cell membrane and actually forces open the pathogen so that the pathogen actually leaks and it kills off the pathogen that way. It's kind of a cool thing. The damaged foreign cell surface for the complement has vasodilators. There are vasodilators. Dilation is when it becomes larger. And when something it vasodilates, remember that also releases histamines. And we've seen histamines before. Histamines are going to be released actually to promote vasodilation. Chemotaxis and opsonization. Opsonization makes invading microorganisms more susceptible to phagocytosis. So they almost kind of like just tag the invading pathogen. They put a little tag on it. And what happens is that signals to phagocytes to come along and engulf that pathogen. That's opsonization. Some cells are triggered to produce interferon. Secreted proteins of virus-infected cells bind to healthy cell surfaces to inhibit virus binding. So the cell will produce interferon. It kind of, uh, puts on its surface a type of protein that signal to other cells that, you know, beware that this virus is in the system, and that enables the other cells to actually protect themselves and possibly build up on antibodies. Just because one cell, not one cell, but you know what I mean, like just a few cells will start to produce interferon within themselves, and it just signals to other cells that there is the presence of a microbe or a virus of some sort. Fever is very important. Remember, we're still talking about the nonspecific defense system. So nonspecific, this is what happens when you get just you know, some sort of injury. This is not at a cellular level, okay? So fever is a good thing. Abnormally high fever is not so good. Anything over 102, 103 for any long period of time is not good. But a temperature is okay. It is a sign that your immune system is working. The hypothalamus heat regulation can be reset by pyrogens secreted by white blood cells. High temperatures actually inhibit the release of iron and zinc from the liver and spleen, and that actually inhibits the bacteria from feeding onto our cells. Fever also increases the speed of tissue repair. You can imagine growing up with me as an anatomy teacher, as a mom, because my kids would get a fever and I would tell them, you'll live. But of course, you have to be real careful. Anything too high. Anything over 102 and above is that's a different story. So a fever is, for the most part, a good thing. And then the final line of defense, this is specific. This is the immune system. So this is the one that is very specific in that it does involve our cells, our 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 internal cells. And um, this is the third line of defense. Not, it is specific. So it does trigger specific cells in our body. And those cells, what we're going to see, are the B cells and the T cells. Antigen specific recognizes and acts against particular foreign substances. So again, we've seen antigens before. We know that we have these proteins on our red blood cells, right? Our erythrocytes. Our body doesn't fight that. At least it should not, for the most part. Our body doesn't fight our own antigen, even though antigens are, you know, basically foreign substances. But if you take my antigen and you're not supposed to have my antigen on my red blood cell, your body will fight for sure. So we have antigens. We've seen that before. Systemic. Remember what I said about the word systemic? We see the word system. So anything referring to systemic is the entire system, not restricted to the initial infection site. And the cool thing about the third line of defense, the immune system, is a lot of times it has memory, recognizes and mounts a stronger attack on the second or the next few encounters of the pathogens. So there are two main types of immunity. Remember, for the third line of defense, this is what we call the immune system. This is at a cellular level. The two main types, humoral and cellular. So humoral immunity, the body's fluids such as interstitial fluid, antibody-mediated immunity, cells produce chemicals for defense. So what you need to remember here is for the humoral immunity, as part of our immune system, two main parts. The humoral immunity, this is referring to our body's ability to make antibodies. So humoral is referring to our body's ability to make antibodies. And one of our lymphocytes that makes antibodies are the B cells. So when we're talking about humoral immunity, what you need to remember is that this is our body making antibodies. What cells make antibodies? Our B cells do. Those are B cells. Those are B lymphocytes. And then cellular immunity, cell mediated. So this is really, really intricate. We're talking mostly about T lymphocytes. T lymphocytes. And this is really, really specific in that the cells target virus-infected cells. I mean, anything from making the infected cell blow up, apoptosis, all the way to tagging the infected cell so that other cells can recognize it. I mean, just all sorts of beautiful, beautiful things going on here. All right, so let's go back and remember antigens. Antigens technically are considered non-self, any substance capable of exciting the immune system. In other words, making the immune system go, hey, we got a problem here. So it provokes an immune response. So antigens really are, they're foreign proteins, foreign nucleic acids, large carbohydrates, some lipids. Super familiar with this one. Lord have mercy. Pathogens, microorganisms. So the antigens, those are the the bacteria, the viruses. So you can see the virus here, this antigen's bacteria. And what happens is what happens when we're exposed to antigens, our bodies can make antibodies that match up with a specific antigen, like a virus. And remember what I said, which cell of our lymphocytes make antibodies? Our B cells do. Self-antigens. Human cells have many surface proteins. Our immune system cells don't attack our own proteins. So, do we have self-antigens? Absolutely. That's what we were just talking about on our red blood cells. We have antigens. Your body recognizes that as self. But then you have these antigens that our body recognizes as non-self. In other words, those antigens should not be there. Those pathogens, in other words, should not be in our body. Those microorganisms should not be in our body. So your body should have the ability, and most likely does, have the ability to tell the difference between whether or not an antigen is self, right? Like on our red blood cells, or non-self. Um, Bacteria, viruses, pathogens. Allergies, many small molecules called heptins or incomplete antigens are not antigenic but link up with our own proteins. The immune system may recognize and respond to a protein heptin combination. The immune response is harmful rather than protective because it attacks our own cells. So this is basically what's going on with allergies. The cells of the immune system. So we talked about this a moment ago, but let's go back and refresh a little bit. The lymphocytes and the macrophage. Remember where we saw these two before? We saw these inside the lymph nodes. We saw these deep inside the medulla of the lymph nodes. These are on the cortex of the lymph nodes. So we have immune cells inside our lymphatics. This is why I teach, pretty much everybody will teach the lymphatic system with the immune system because they really go hand in hand. So the lymphocytes, remember the two types of lymphocytes. Also remember that lymphocytes are leukocytes. In other words, lymphocytes are white blood cells. The two types of lymphocytes, B cells and T cells. Cool beans? They originate from hemocytoblasts in red bone marrow. B lymphocytes become immunocompetent in bone marrow. What this means, immunocompetent, means that they they mature in our bone marrow. So you can remember B lymphocytes, bone marrow. That's where they go off to mature. Okay. T lymphocytes, they become immunocompetent in the thymus. So these cells will travel into the thymus. Some will travel into our bone marrow also, and that's where they mature. Macrophage arise from monocytes, become widely distributed in lymphoid organs. Remember the macrophage, they consume the pathogen via phagocytosis, where they engulf it and disintegrate. The difference between B cells and T cells. I don't even know where I found this. B cells, I attack invaders outside the cells, and the T cells attack invaders inside the cells. That's kind of cool. And they're showing us on the B cell, recognize this as an antibody. So B cells make antibodies. T cells are just phenomenal. This is a receptor protein, and basically it just allows cells to recognize that there's a pathogen, there's a microorganism in the body. All right, let's go back to the humoral. Remember we said humoral? That is referring to the B lymphocytes. And we said B lymphocytes make antibodies. All right. So remember our two types? This was the third line of defense, humoral, and the other one, remember the other one was cellular? So humoral antibodies, these are the B lymphocytes. The cellular, those are the T lymphocytes. All right, so the B lymphocytes make antibodies with specific receptors that bind to specific antigens. The binding events activates the lymphocyte to undergo clonal selection. A large number of clones are produced, primary humoral response. That's cool beans. And we actually have just hundreds of thousands of different types of antibodies that our B cells can make. Most B cells become plasma cells. Yes, we are talking about plasma. Remember we said that was about 55% of what what makes up our blood. So B cells become plasma cells. And so that's pretty cool. What happens is when the B cells turn into plasma cells, which is blood, right? The other part of our blood, other than our red blood cells, that's traveling through our system. So these antibodies would then travel through our system. Pretty darn convenient, right? That's cool beans. Some B cells become long-living memory cells. And this is showing how the B cell does turn into, it just kind of secretes all these. I mean, it just depends on what the body's experiencing. There can be millions of different types of antibodies. And it does have the ability to form a memory, a secondary response. So the second time the body is exposed to that same pathogen where our body already formed a memory to it, so there's already the antibody had been made by the B cells, the next time your body is exposed to it, you have a secondary response. You don't get as sick. In fact, a lot of times you may not even know you have that pathogen because your body deals with it so quickly. So again, what do your B cells do? When they encounter an antigen, they will produce antibodies. There is also passive immunity. Antibodies are obtained from someone else. You'll see this from mother to fetus, mom to baby. Immunological memory does not occur in this case, but a mother can pass antibodies to her infant to protect it for the short run. Protection provided by borrowed antibodies, in other words. So this is called passive immunity. We'll find it mostly between mother and fetus, mother and baby. Antibodies prepared for clinical testing or diagnostic services produced from descendants of a single cell line. So the monoclonal antibodies, this is just making colonies of antibodies. They used to do this for pregnancy tests. They don't do it anymore. And it can be a type of treatment if a person has been exposed to a type of hepatitis, one of the types of hepatitis, or rabies. Antibodies. Now, antibodies, the real name of antibodies is this big old long protein name. Remember, you guys, the antibodies are our fighters, okay? These are the good guys. The antigens are not so much the good guys. So the antibodies, the big old long fun name that you can impress your friends with, immunoglobulins, or IGs. All right, soluble proteins secreted by, remember what cell makes antibodies? Our B cells do. And some of those B cells, some of those antibodies can turn into plasma cells. Carried by blood plasma, that's cool beans, because blood plasma, that's the blood, y'all. So those antibodies travel through your blood. That's cool beans. Capable of binding specifically to an antigen. And this is what the antibody looks like, basically. And so it has a heavy chain, kind of shaped like a big old Y, 4 amino acid chains linked by disulfide bonds. So we can see the disulfide bonds. And at the very end of the Y, at the very tips up and through here, these are the antigen binding sites. This is a lock and key. This antibody right here fits specifically to a certain type of antigen. It's a lock and key. So we'll see the change. Remember I said there are millions of types of antibodies. So the key right here is the very end of the Y, depending on how it fits to a specific antigen. And it just kind of locks onto that antigen and destroys it, or it builds a memory. It is very, very specific. And I think I have some examples. So you can see the lock and key format here. Oops. That's kind of a neat little view of, oops, did I miss something? Nope. Kind of a 3D view of an antibody. Different classes of antibodies. There are 5 major antibody classes or immunoglobulin classes. IgMs, can fix infections. When you see fix, it means it attaches to the infection, the pathogen, the microorganism, whatever, the bad thing, the antigen, and it fixes onto it, and it kind of breaks it down. IgAs found mostly in mucus. So these are antibodies found mostly in our mucous membranes. IgDs, they're important in the activation of B cells. And then IgGs can cross the placental barrier, so we'll find the IgGs mostly in during pregnancy. IgEs, these are the ones involved in allergies. And of course, on a daily basis, I say my IgEs are going insane because I have horrendous allergies, especially this time of year. So those are involved in allergies. And remember, so the antibodies, they have a different kind, a basic shape, right? They all have, so here's an IgA. They have the basic shape, but there are just areas on, especially at the end of the Y, that really denotes it as being a specific type of antibody. The IgMs are huge, right? IgMs are the big antibodies. Antibody function. Antibodies inactivate antigens in a number of ways. So antibodies, where do these come from again? What lymphocyte makes antibodies? That's right, B cells. And what they're going to do is they're going to inactivate antigens. Remember, the antigens are the bad guys. Fighters, bad guys. So a couple ways that they can stop the infection: a complement fixation, neutralization, agglutination, and precipitation. So these are some ways of how antibodies can function. And this is just the tip of the iceberg, y'all. I'm just going to kind of overview it, okay? So neutralization, this is an example of neutralization of an antibody attacking a virus. We can see the virus in there. I bet you that's COVID right there. That's COVID-19. I can see. I'm kidding. So neutralization, mass dangerous parts of bacteria, exotoxins, and viruses. Yep, there it is. That's the COVID-19. Agglutination, you can see the antibodies clumping together the cells. Precipitation, these are soluble antigens. And remember I told you earlier about complement. I said that complement is just a group of antibodies that form on the membrane of the pathogen and basically just, it's almost like if you let the air out of a tire or something, or out of a balloon, how it just kind of the air seeps out. So these complement actually form, they just puncture through the membrane of the antigen, and it just kind of leaks out. That pathogen just kind of leaks out. Kind of cool stuff. Phagocytosis we have seen before. Inflammation, yes, the release of histamines, vasodilates, and causes swelling. And sometimes antibodies can sometimes just make the cell blow up. That's called cellular lysis. I want to show you one thing. Wanted to show you one thing real quick about the antibodies. And let's take a listen. So this is a brief video on antibodies.
Antibodies are proteins that play an integral role in the complex system of specific immunity. They bind to and inactivate foreign proteins called antigens. Each type of antibody binds to one kind of antigen molecule. B cells are one of the two types of white blood cells that make antibodies. When a bacterial cell enters the bloodstream, proteins on its surface act as antigens and trigger response by the immune system. These antigens will bind to the unique B cells that have the corresponding antibodies on their surfaces. These B cells now divide. Some continue to divide, producing many plasma cells. Plasma cells secrete large quantities of antibodies into the bloodstream. This division of only the appropriate B cells is called clonal selection.
All right, cool beans. So, where are we? So, let's go ahead and go into the second type. So, our first, oops, hello. Can we play from current slide? Thank you. So, our first type, that was humoral, right? When B cells make antibodies. The second type of immune response is cellular, cell mediated. So this is really down, this is at a, and not to sound redundant, but this is at a cellular level. This is the nitty gritty. So we're talking about cell mediated immune response focused mostly on the T cells, the T lymphocytes, okay? So antigens must be presented by the macrophage to an immunocompetent T cell. So it's basically, I kind of think about, just here is this macrophage saying, here, T cell, this is the bad guy, you need to do something about it. I kind of think about the Lion King where they held up the little baby lion and said, here it is. I kind of think about that situation, but just kind of is just presenting the antigen to the T cell. The T cells, you guys, they're the boss, okay? Especially the T helper cells. Those guys, I call them the generals because they are basically in charge of all the other lymphocytes. So these guys are the bomb, okay? So what happens is the macrophage will come up to the T cell and say, look, here is this antigen. You need to do something about it. Just presents it. So antigen presentation. T cells must recognize the difference between self and non-self, right? Remember self? Um, is the T cell going to attack your antigens on your red blood cells? Well, I certainly hope not. It should recognize the difference between what's ours and what's not ours, right? This is the pathogen, right? This is the bad guy. After antigen binding, clones form as B cells, but different classes of cells are produced. Two cells involved in cell mediated, the macrophage and T cells. I mean, this is pretty darn specific, y'all, okay? So the T cells and the macrophage are really involved mostly in cellular mediated immune response. And this is a really good example. So if you take a look, the brown here is representing a macrophage. Remember, the macrophage consumes via phagocytosis. Do you see how it's consuming the antigen, the virus? Consumes it, breaks it down, and what happens is the macrophage takes a little piece of the antigen. It's like, you know, puts it on its outer membrane, and basically says, "Here, I destroyed it. Here it is. Come look." And along comes the T cell. And remember what I said, you guys? The T cells, they're the bomb, man. They are the general. They rule all the other fighter cells. Okay, especially the helper T. They are the boss. So the helper T comes along and says, "Ah, I recognize that. Thank you for presenting that antigen." So the helper T cell says, "Okay, we know what it looks like now," and it just can go out into our body and then kill off the other antigens. That same antigen, because it got a little taste of it right there, got a little bit of a presentation, and it does that by the B cells. Remember what we said about B cells, antibodies, or it can do a cytotoxic killer T cell. That's right. This is cell mediated. So this cytotoxic killer T cell, cyto is cell, toxic, well, it's the killer. All right, these are the bad dudes. Okay, do you see how cool that is? So it just kind of puts this nasty little, hey, I killed this thing. Let me show you. I don't know if you've ever had your cat bring home a mouse and say, it brings it to the back door and shows you a mouse that it caught, a dead mouse. And it's like, look, mom, look at what I brought you. That's kind of what's going on here. It just takes a little bit of the antigen, puts it on its membrane. Helper T comes along and says, got it. I can have that memory now, and now I know what to do. Tell the B cells to make antibodies or make cytotoxic T cells. Cool beans. Three types of T cells. Cytotoxic T cells specialize in killing infected cells. Insert a toxic chemical. The toxic chemical is called perforin. The helper T, those guys are the bomb diggity, okay? The helper T, they're the boss. Recruit other cells to fight the invaders. Interact directly with B cells. You remember what the B cells are? The B lymphocytes? What do they make? That's right. I know you said antibodies. But these guys are the bomb, okay? They basically tell everybody else what to do, all right? That's what the helper T cells do. And then some T cells are called suppressor T cells. So we don't want the system to keep producing, cytotoxic T cells because that's not necessarily a good thing, especially if the pathogen has been destroyed. If you keep having these cells being produced, that's not a good thing. It can really have a bad reaction in your body. So, what does your body do? Amazingly so, it has suppressor T cells, and that stops the whole process. So it's more controlled. So the suppressor T cells release chemicals to suppress the activity of the T and the B cells. Brilliant, right? Brilliant. Stops the immune response to prevent uncontrolled activity. A few members of each clone are memory cells. So sometimes T cells can also, like B cells, we said B cells have memory. Well, so do T cells. Helper T cells, the boss, see how they're the general? They're also known as CD4. They express the CD4 protein on their surface. They're presented with antigens. This is a group of cells and secrete cytokines that regulate or assist in the immune response. So these helper T cells, they tell everybody else what to do. A little bit of a summary. So our two main types of immune response. Do you remember what stage the immune response is? Is it the first line of defense, second line of defense, or the third line of defense? Ah, you're right. I know you said third line of defense. Good for you. So the immune response is the third line of defense. Humoral, that's where the B cells make antibodies. Cell-mediated, those are the T cells. Cool beans? It's a nice little slide. What about organ transplants and rejections? Major types of grafts: autografts, tissue transplanted from one site to another on the same person; isografts, tissue grafts from identical persons, identical twins; allografts, taken from an unrelated person; and then, of course, xenografts. These are taken from an animal species. Autografts and isografts are ideal. Xenografts are never successful. Allografts are more successful with a closer um, tissue match. Disorders of immunity. So we already said allergies. Oh boy. Now, some of us have allergies all the time, but some allergies will kill you, right? This is if you have a cellular type of allergy, like you are allergic to bee stings, if you're allergic to peanuts, I'm not just talking hay fever, I'm talking at a cellular level. It's pretty severe. Immediate hypersensitivity triggered by the release of histamine. There are the IgEs, right? The immunoglobulin type E of the antibodies binding to mast cells. Reactions begin within seconds of contacts. Sometimes, you guys, it can be so dangerous, it can kill. We call this anaphylactic shock. That is a bad, bad thing unless the person gets treatment right away. I used to know some kids that couldn't even be in the vicinity of a peanut butter sandwich because even in the air they would go into anaphylactic shock. So these severe allergies are nothing to laugh at. I mean, these are pretty serious. Delayed hypersensitivity triggered by the release of lymphokines from activated helper T cells. Symptoms usually appear 1 to 3 days after the antigen. So some happen quickly, in other words, some happen over a period of time. And here are some examples. You can see this person was allergic to probably the plastic or whatever the shoe was made out of, the flip-flops. She's having, Riley, hush. She's having an allergic reaction. You can see the swelling, just absolutely frightening. This girl, this is actually the same girl, and she is having, I don't know what type of reaction they're having, but they are definitely having an allergic reaction. So the antigen invades the body. Remember what I said about antigen? That's the bad guy. The plasma cells, remember where the plasma cells are coming from. Those are B cells, right? Those are B lymphocytes. Produce large amounts of antibodies. If it's an allergic reaction, what type of antibody? IgEs. The IgE antibodies attach to the mast cells in the body tissue and to circulating basophils. Hey, we haven't seen basophils in a while, right? Secondary response. Sometimes a secondary response, the second time of exposure, can be even more severe. I know personally my allergic reactions to pain medicines happened over a period of time until today, where I can't even take pain medicine or it'll kill me. So sometimes the second and the subsequent exposure to the antigen it's much more severe each time. Disorders of the immune system, immunodeficiencies, production or function of the immune cells or complement is abnormal. Remember what I said about complement? That was that group of 20 proteins that kind of they puncture the membrane of the virus, the antigen, and kind of bleed it out. May be congenital or acquired. This includes AIDS, acquired immune deficiency syndrome. The immune system does not distinguish between self and non-self when we are talking about autoimmune diseases. So one very serious disorder of the immune system is the autoimmune disease. Body doesn't, you will hear people say that the immune system is where the body fights itself. That's very true. But why? It fights itself because it can't distinguish between self and non-self. In other words, it can't distinguish between what's supposed to be there and non-self, that thing that's not supposed to be there, like the pathogen, right? The body produces antibodies and sensitizes T lymphocytes to attack its own tissue. Some examples: multiple sclerosis, white matter of the brain and spinal cord are destroyed. Myasthenia gravis impairs communication between nerves and skeletal muscles. Juvenile diabetes destroys pancreatic beta cells that produce insulin. Remember we said the beta cells, what they produce? So the juvenile diabetes, this is the one that you're born with. You know, these people are born with this. They don't have beta cells. Rheumatoid arthritis is also an autoimmune disease. Lupus is an autoimmune disease. Glomerulonephritis, nephritis, anytime you see the word nephro, it is referring to the kidneys. So this is impaired renal function. Self-tolerance breakdown, inefficient lymphocyte programming, appearance of self-proteins in the circulation that have not been exposed to the immune system, for example, egg sperm and eye lens. Sometimes there can be a cross-reaction of antibodies produced against foreign antigens with self-antigens. One good example of that, where there was this crisscross of antibodies to where the body should recognize it as self, but then it's confused and thinks it's non-self. Another real good example is rheumatic fever. It actually attacks the heart. And then the developmental aspects of the lymphatic system, body defenses, except for the thymus and the spleen. Lymphoid organs are poorly developed before birth. A newborn has no functioning lymphocytes at birth, only passive immunity. Remember we said from mother to baby? If lymphocytes are removed or lost, severe edema may result. Remember we saw lymphedema earlier. Edema, that's when the surrounding tissue can't be drained very well. And let me show you this real quick. So remember, this is kind of just an overview of the lymphatic system?
Lymphatic vessels recycle excess tissue fluid that accumulates in the interstitium as a result of normal capillary leakage. The walls of the lymphatic vessels act as one-way valves, only letting fluid enter. Once in the vessels, the lymph is advanced toward the systemic circulation. Along the way, the lymph is filtered through several lymph nodes where it is inspected for foreign substances. Upon reaching the thoracic duct, the lymph re-enters circulation.
That's cool beans. And I think I wanted to show you just one more antibodies and the immune response. Let's take a look at this one. This is the immune response. I believe this one I'm showing you is the T cells.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Bravo. Yay, us. All right. So I believe that was the end of our lecture. And I don't know where I am. There I am. I'm right there. So watch the video. If you have questions, watch the video again, or shoot me a text message, answer your guided notes, and I'll let you know when to upload those. And there you go. That is the immune system and the lymphatic system. I hope that you learned a lot today. And be sure to watch the two videos, part 1. This is part 2. And thank you very much. Bye, guys.